Martin, thank you for the introduction. So I'm a little surprised. I didn't expect so much like this, audience of this large population. So I really appreciate it. Thanks for coming. And I believe the Zoom people could hear me, right? So okay, great. So so today I'll be talking a little bit about my projects, which is to detect locations and dynamics of ligand-receptor signaling at cellular resolution from spatial transcriptomic. It is a really long topic, but I guess it says the main idea clearly. Just we do cell-cell communication, and it's from spatial transcriptomics. So let's start from the background. So cell-cell communication is vital for all kinds of biological processes at different stages of the cells. And all multicellular organisms rely on intercellular communication networks to coordinate any kinds of the physiological functions. And these interactions involve structures and functional proteins, small compounds, and extracellular matrix, membrane proteins, and extracellular vesicles, all kinds of mechanisms. But however, we do think that the mechanism ligands that bind to required receptors on the other cells, or what we call like the ligand-receptor interactions, are the dominant mechanisms used for cell-cell communication inferences. And if you ask me, then there are already a lot of cell-cell communication methods on the market. So there's really like most of the methods listed here are enabled or inspired by high throughput experimental methods. For example, the use of the single-cell RNA-seq allows reviewing, which ligands and receptors are expressed in which cell, or in each cell, or in each cell type, or a cell within heterogeneous tissues. So this is a really nice plot I adopted from the Nature Genetics Review paper. And more, but if we think about that, so there are several main, disadvantages. So first of all, we believe that most methods do not incorporate real spatial information. And second, they only infer signaling among groups of the cells rather than the cell-to-cell resolutions. And finally, they do not infer the dynamics of the cell-cell communications. So here, in order to solve these challenges, we present CytoSignal and Medial CytoSignal. So one of our key insight here is we believe ligand-receptor signaling is driven by a protein-protein interactions that occurs at a specific location in the tissue. And we want to use spatial transcriptomics now enables, of course, you measure the transcriptomics and also their corresponding locations in the tissue. So we want to use that to predict the amount of ligand-receptor protein-protein interaction at each position in the tissue. So so the key idea. So if we're talking about the algorithm here, the key idea is simply we believe a cell-cell signal interaction only occurs when all of the required ligands and receptors components are expressed in a close spatial proximities. So I guess the key assumption here, or if we address it in other words, is we believe cells only communicate with its real spatial neighbors. So there's no way that, one cell could communicate, for example, in the mouse embryo, you couldn't assume one cell in the, some part of the tail, it commutes directly to some cells in the brain. It's just not possible. So this is why we we assume this, let me see which is the laser point. Okay. So this is why I assume so we kind of infer the gene expression in neighborhood. And, since communications are mediated through different ways, we define this spatial neighborhood differently for two classes of the signal interactions. There's the contact-dependent interactions and the diffusion-dependent interactions. And for contact-dependent interactions, we only define the neighbors as the directly connected neighbors, which means one cell, so only the cell that can directly touch the index cells are these close neighbors. And on the diffusion-dependent ones, we assume a 200 micrometer radius diffusion radius, which means we believe the index cells could send ligands that travel through approximately 200 micrometers within the tissue to reach the other cells. And of course, so the ligand and ligand receptor structures could be pretty complex. So we do consider like, different subunits in the ligands and the receptors. And so next, CytoSignal constructs like null distributions to give the statistical power to our inferences. And we then calculated, a one-sided p-value with a spatial FDR for the null hypothesis. To calculate a one-sided p-value with spatial FDR to infer to test whether our null hypothesis is true or not. So our null hypothesis is the signaling strings observed for a particular cell is no larger than expected based on ligand receptor expressions. And finally, we also we calculated, and from there, I have to apologize, I didn't practice for this, and it has been some time since I presented for the crowd. So since we have like a score for each interaction in each cells, we can easily identify some pretty obvious signaling patterns. It's similar to sometimes you see patterns of a gene that express only in a specific region of the tissues, and we can see the same thing for the interactions. And finally, since we have this cellular resolution scores, we can infer this interaction-related genes, because I believe like what the biologists care the most is if you tell me interactions, I would hope you could tell me a bit more about what genes are related to that specific interaction. And we enable that through a sparse regression. Let me show some results here. So, already have the algorithm, and it's interesting if we can, and we want to definitely apply it on different kind of systems. So we first applied it on the slice-seq, on the mouse embryo data, on the mouse embryo cortex captured by the slice-seq data set. So slice-seq is a cellular resolution spatial transcriptomic technique. And you could see here we these are some of the mean outputs of CytoSignal. So in the first plot is a heat map where each point represents a score for a specific interaction in that position. And then the second one is the significant, since we have a statistical test, we can then know which cells are truly having significant interactions, significant signaling activities. And then we came up with a really novel like edge plot, which tells you, you could see this like thin lines that connects the cells in the bottom panel to the top panel. That just means, it just tells you like which cells are roughly communicating with the index cells here. And since we have, the cellular resolution score, we're then interested in finding maybe some potential spatial pattern. And then we decided because we're using this cortex system, we find a really interesting thing is, if you plot the scores along the depths from the most outer layer to the inner layer of the cortex, you could see a really interesting pattern. Like the signaling, the scores actually increases first and then drops, and it reaches its highest in the SVZ layer, which are very interesting in the brain development. And of course, we applied it on another cortex, which is captured by slide text. It's another single cell resolution spatial transcriptomics from the same lab, actually. And we actually see the same thing. You could see like the cells are, for this specific interaction we choose, the cells in the radial glia are having significant activities. And if you plot the scores along the distance from the centroid of the cortex to the edge, you will also see a very interesting line. It basically says the signaling intensity increases from the edge all the way to the radial glia and reaches tops and then drops again. And since CytoSignal models, diffusion-dependent interactions and contact-dependent interactions separately, our next question will be, if you can model them separately, what will be, could you identify some differences from these two categories? And here is we applied a CytoSignal on a mouse embryo atlases measured by stereo-seq. It has data set from all the way from E12.5 to 16.5, and we applied it on each single slide of the data. And then so here's a heat map summarizing all the 200 diffusion-dependent interactions at each spot. Because like here, very interestingly, we see like the diffusion interactions seems to be very enriched in this Jiao and T's region, and compared to the contact-dependent interactions. So it suggests maybe the diffusion-dependent interactions are dominating during the Jiao and T's bone development. And we also identify another example in this time, which is the contact-dependent interactions outperform the diffusion-dependent interaction. With this time is in the brain, in the mouse brain. It's the same data set, which is focused on different regions. You could see like the contact-dependent interaction has a really interesting. It kind of depicts the border of the brain and the meninges. I mean, especially I think the purple colors are the the purple clusters are the chorial plexus. The orange are the brain, and the yellow are the meninges. You could see like they kind of depict the border regions, the intersection regions between different clusters. This is really interesting. And another advantage of it's just like another key challenge in all of these cell-cell combination studies is, of course, the validation. Like you see, like even if you say, okay, we saw like this interaction is significant in what kind of tissues, you will never be 100% sure unless you validate it in the real tissues. So this is what we come up with this strategy. So we use like by this proximity ligation assay, which is basically a fluorescence technique. Since we have ligand and how to say, so it specifically it's like so this technique involves the use of two primary antibodies. So in our case, we have ligands and receptors, right? So we have primary antibodies, one of them on the ligands, another on the receptors. And once these primary antibodies come close together, or specifically it's like less than 40 nanometers, it will give you a red fluorescence signals. So so therefore, like in the context of ligand receptor bindings, we think like this PLA technique is wonderful. It provides direct evidence that whether the proteins are really co-localized or they're really close to each other on the cellular surface. So here's some. I mean, of course we have it. We have done it on the mouse embryo data, and we think it's this is E12.5 mouse embryo data. And you can see on so here is like, you see like this column is from the only the first column is the R score inferred by CytoSignal, and the other two are from the PLA fluorescence images. You could see there's a really good line between our inferred activity and the the true activities identified by PLA. And you could see roughly the same for another interaction we picked within the brain region. And also show like the false positives. This is where I didn't show on the plot, but this is where the other cell-cell communication methods indicate like there should be a signal over here, but if we use CytoSignal and PLA, there's no signal. So just perform, it identified the false positives. And we have so the previous slides is about the contact-dependent interactions, and also have we test a few more from the diffusion-dependent interactions. And this time we basically see a same story. We're seeing it's giving this good correspondence and identifying false positive from previous methods in different regions, in different areas within the embryos. And finally, since Another actually important benefit of CytoSignal is it enables the identification of differentially expressed genes associated with with each signal interactions of interest. So because we have because it's just like we have for each interaction we have a score on each cell. We can do a sparse regression test here, and gives you which genes are contributing significantly to predict VLR scores. And from there, you could just pass those genes to the downstream analysis. So here, take the goal analysis as an example. You'll see like it gives from for these interactions that's enriched in the radioglare region, it highlights a few genes and which is closely related to the neural system development and the brain development. And another key challenge in the studies of cell-cell communication is how does this signaling interaction change over time? So it is, people like previous methods often only tells you, this is what we see in the data, and we believe this is happening right now. But the next question will be, okay, how does it change over time? So this is how we came up with Weizhou CytoSignal as an extension. Is like if we are, it's by combining RNA velocity, which is RNA velocity, or in other words, VelVE from our lab, is another software we developed for inferring the RNA velocity of the cells. By combining RNA velocity and CytoSignal, we are able to see, whether each interaction is increasing, or the activity of that interaction is increasing or decreasing directly mapped to the tissue plane. So here is an example. It's like we identify. So here we show an example of the interaction called ALBU FcRn complex, which is specifically enriched in the liver. So this interaction is a well-known. It has tons of literature support. It's just like so ALBU protein is only secreted by the liver, so you couldn't see it elsewhere in the tissue. And by having these arrow plots, so these 3D arrow plots, the red arrows means it's increasing, and the length of the arrow just means how the extent of how dramatically it's changing, and the blue arrows is indicating it's going down. The activities will be decreasing. So you can see it's really it highlights across four time points. It really highlights this extremely increasing activity in the liver. And here's like some arrow plots for each in the dynamics of each individual genes. And another example will be this Win5A ANT-XR interactions in the gel in gel and Ts. So Win signaling is also another really famous, really well validated signaling pathway during the bone development. And again, the story here is you see there's really a dramatic increasing trend in the gels and Ts area across different time points, which kind of validates our finding is true. So finally, CytoSignal it's it's just so one another important features of any kinds of computational tools is its adaptability. So if you can even if in the paper you show you could use this data, you could use your algorithms on one of the data, there will always be other researchers come and ask, okay, can we use your data? Can we use your algorithm on other kinds of maybe 10 other data sets? And CytoSignal has really good advantage of it's almost compatible with every spatial transcriptomic techniques on the market right now. So here's an example. If we apply CytoSignal on the Visium HD data, and we don't even need to do deconvolutions. 10x Visium HD data is it has a higher resolution it's a little bit higher. I think its resolution is about 100 micrometers. As a comparison, each single cell is around 10 micrometers. So people usually process 10x Visium data first with the deconvolutions, and then and then you could do all the downstream one. But CytoSignal does not need to do that. You just apply it directly on the data. And another example is like Murfish. This is also another classic. This is Murfish is another is a like fluorescence probe based technique, which is a little bit different from 10x Visium, which is based on barcode capturing. But we can still do it and identify some really interesting signal enrichment in the LV LV ventricles in the brain. And of course, another example is like StarMap Plus. It's another fluorescence-based protocol, and we identify some really interesting thing there. So this one is it's for all the so this one is like we really want to see whether CytoSignal outperform all the other masses on the field. So here we benchmark CytoSignal with three really famous cell-cell communication software: CellChat, CellPhoneDB, and CellChat Spatial. So here is just like a line plot of highlighting the statistical significant. So this is on the x-axis is the distance between the cells that are interacting, and the y-axis is the density. You could see CytoSignal do not consider cells that are more that are far far away than 200 micrometers to be significant, while the other softwares you could see it's really is like a hill. So it gives you even tells you like cells that are 8,000 micrometers away are significant, which is totally not reasonable at all. So the takeaways of my presentation will be we have 6 advantages. We always hope people to remember our advantages. So these are kind of the experiments you could kind of the analysis you can do using CytoSignal. We have a simple interpretable score at each position in tissue, and it separately quantifies contact-dependent versus diffusion-dependent interactions. And we can validate using antibodies, and we can identify spatial gradients, and we can infer the related genes for each ligand-receptor interactions. And finally, you can identify the temporal changes, or the how does the signaling changes across over time. And Martin already advertises our paper, and really appreciate it. So CytoSignal is public right now. We have it on GitHub, we have it on bioRxiv, so feel free to check it out. And thank you for your listening. I'll be happy to take any questions. Yeah. Oh, please.
So since you're able to use this on Visium spots, does that mean since those spots are about 100 micrometers, and then you're saying like even was it 200 micrometer radius, and then contact, I assume is that just like a smaller radius, or do you need like segmented cells for that? Like how are you using like for transcript or segmented cells?
Yeah, that's a really good question. So I believe for usual, so you're talking about Visium data, not Visium HD, right?
I guess kind of more broadly, just are you the data that needs to be input, you need segmented, or like how are these neighborhoods generated?
So, yeah, so I guess so for using CytoSignal, all like the input data will be only you have like a cell by gene matrix. So that cell does not need to be segmented cell. It could be like a a position or a grid, depends on how like what your data is. It's just like but for Visium data, which has 100 micrometer resolution, so that's basically a spot instead of a cell, right? It usually have like more than 20, 30, or even more cells there. So just like so I guess you can definitely use that. It's just like our assumption does not. I think even for those kind of data, our assumption did not change. So for contact dependent interactions, you just don't need to find the directly connected neighbor anymore, because you already have a lot of cells in each spot. So you could just run without imputation. But for diffusion dependent interactions, even, a lot of cells in one spot, like the protein could still be diffused to the other spots. So you could just, apply it, and use CytoSignal. Yeah.
So just kind of the interpretation is a little bit different depending on your input data.
The interpretation might be a little bit different, depends on your data, but but it's definitely usable, and it's CytoSignal supports that technique. So, yeah, no problem. Please.
Great talk. Ideally for the 3D structures, or for the distance between different cell type, when they become close or their distance is very close to each other, their expression level supposed to be expression correlated, or expression patterns trend to be more similar. Have you tried to summarize or do the similar analyze?
I'm not sure I understand your question.
My question is, when different cells they have interaction, or they are very close to each other,
Yeah.
their expression level or expression patterns should be correlated with each other. They have the similar 3D structures close to each other. Their expression should be similar. So have you ever tried to comparing the two important values for your data?
So are you asking, like, do we consider, the similarities between cells in CytoSignal?
Yes, and and for the distance and for the expression, the similarities for each other.
So what we are, so I guess it's related to how we do, how we infer the proteins are, diffused through the space, right? We basically use a Gaussian kernel to, transfer the distance between the cells to similarities, and use that, like, it's similar to a weighted sum across the, for the expression levels. Yeah, so. What was that?
Later.
Oh, okay. Please.
I was wondering about your performance tracking, where you compare with the cell and DB stuff.
I mean, this one? Oh, okay.
Maybe I could be wrong or the process. Does cell chat with cell and DB take into business into consideration at all? I wonder, like, you just draw a cutoff in the inverse distance, so that's why you consider cells far away from a certain distance. That's why you have this off-target predictions. But for them, I guess cell set spatial maybe already taking into that consideration. So I guess I have two questions. Why is that? How did you draw this performance tracking? And the second is, like, what is your advantage taking distance into account in your system compared with other, like, for example, cell chat spatial that makes your algorithm different better?
What is your first question again? Sorry.
Like, does the cell with cell chat did you actually take into spatial or distance information into consideration at all?
You mean they consider that or not consider that?
Yeah. As far as I know, like, do they? I'm not
so
sure,
yeah, so it's like I believe cell phone DB and cell chat, like, they have a lot of versions right now. I think cell phone DB have maybe 6 versions. So I think this is version 2 or 3.
Okay,
So I think this version do not consider spatial information.
Okay.
But you can see that for cell chat here, we do consider, we include 2 versions. So cell chat, I think it's on the right upper corner is the version 2 that do not consider. And it's related to a second question. So weirdly, even with spatial information considered, they still having a lot of false positives on the the right tail. And this is what we want to highlight. It's like, just like in both cases, there are a lot of false positives. Yeah,
that's just like from a technical perspective, what's the advantage, like the way that you take spatial information into consideration that makes your that's more accurate.
So it's like, so CytoSignal is considered like CytoSignal does, like, we do our inference at the cellular level. Just like we actually, we construct all of our statistical tests for each cell. So all the other three methods here, cell chat, cell phone DB, and cell chat spatial, they are all at, I guess it's cluster level. So that's why they're having, positive because their resolution much lower. So yeah, I guess that's the key.
That makes sense there, yeah. Thank you.
Please.
We kind of follow up to that. So if you looked at their kind of cell chat or stuff on DB, you could set a threshold like you have with yours at 40 and 200. Have you compared your like direct results within that kind of specific defined area to see how similar your results might be compared to theirs?
I think for cell chat spatial, we do set that directly. I mean, the same directly 200 micrometers, and it's still giving us results like this for some reason.
Yeah,
So-
but this is like a big overall kind of summary of how many interactions you had. But like looking at an individual summary of this cell is
You-
interacting with so many neighbors, are you finding that it's a similar kind of population you're finding within cell chat?
Oh,
Other than the fact that they have more dispersed neighbors as well, but you set like a upper bound of 200.
I mean, since cell chat spatial is at cluster level, so they're only telling you which cluster is interacting with each cluster. So there's no way to say,
Okay.
okay, maybe only a portion of the, and actually that's another advantage. If you look at the, some of our actual results, we're only, we're basically only telling you a portion of the cell type is interacting another, maybe some portion of another cell type. This is a really good advantage. Like the cell type, well, cell chat only tells you, SBZ is interacting with BZ and that's all. There's no, zoom in resolution of the communication events. Yeah, thanks. Do I miss any questions online?
Nope, nothing online at the moment. Any more in the room?
I have a question, short question.
Please.
So you are aware of Medium HD, right?
Image, yes.
So when apply a type similar to Medium HD, is there any difference, of course, difference in size?
Oh, so, I mean, there definitely will be difference because, VZM HD gives you three resolutions, right? 2 micrometers, 8 micrometers, 16 micrometers. So below 10 micrometers is usually considered as subcellular. I don't think CytoSignal is applied, it's compatible with the 2 micrometer resolution because we're not making any assumptions with intracellular communication, which means, the communication going within each individual. So that's another topic. But I think for 8 micrometer and 16 micrometer, there should be similar to the other single cell level spatial data we use.
You guys speak out?
Thanks so much.
Go on.